Students, so today we will be doing about the factors affecting the enzyme activity. We will be doing only one. You have taught in the school that there are two factors affecting the enzyme activity. One is temperature and the other one is the pH. But actually, I taught three of them because there is another factor that affects enzyme activity in the topic of digestion, that is concentration. I will be teaching it together, all the three factors. But only in this video we will be doing alone the pH because it's alone a lengthy topic. So let's just jump on the topic. First factor about the pH. Just erase every memory you have about the topic of pH. Erase every memory. We will be starting from the trash. Just suppose you don't know anything about enzyme or whatsoever. Enzymes, just listen what I said. Enzymes are highly sensitive to pH change. What do I mean by highly sensitive? Highly sensitive, if a pH, if an enzyme, just suppose an enzyme pepsin, this enzyme wants the pH of 2 or 3. If I change slightly, like if I take to the pH of 1 or to the pH of 4, this enzymes thus will affect the enzyme activity. This will denature pepsin and lower down the enzyme activity or permanently damage pepsin. If we talk about another enzyme, just suppose protease, just suppose its enzyme activity is 7, 6 or either 8. It's actually this. But just suppose if I drop it to 5 or if I raise it to 9, will it affect enzyme activity? The answer is yes. It will affect enzyme activity. How? We will be talking about. So what do I mean by this sentence? Highly sensitive. What do I mean by this word highly sensitive? I mean if we drop or raise the pH level of that enzyme, it will denature or permanently damage that enzyme. Alright, the second, slight change from optimum pH can affect hydrogen bonds. So what the heck is the hydrogen bonds? What are hydrogen bonds? Hydrogen bonds are the bonds that keeping the enzyme together. This is an enzyme, just suppose this is a protease enzyme or whatsoever enzyme it is called, I don't know. We talk that enzymes are made of proteins. It is made of protein, thousands of molecules of proteins. Just suppose it's made of protein. Now, what is the thing that keeping these protein molecules together? Why they are together? Why this molecule is here? Why this molecule is not here? Why they are sticking together? Because of the hydrogen bonds. Now, I won't go deep in the detail how hydrogen bond is formed. What is hydrogen bond? Just just memorize hydrogen bonds are the bonds between the protein molecules inside the enzyme. That's keeping the enzyme molecules together. Now what do I mean by slight change? I told you that slight change, even dropping a level of 1 level of pH, like dropping from 2 to 1, can affect the enzyme activity. How? It can affect the hydrogen bonds. If we decrease it from 2 to 1, it will affect the hydrogen bonds that is keeping the proteins together. From optimum, what is the word optimum? Optimum, I told you pepsin has the pH of 2 to 3. This is the optimum level. At this pH, enzymes works at its high level. If we give pepsin this pH, it will show the highest enzyme activity in any other pH. Like if I give pepsin 4, it will show less enzyme activity. If I give this pepsin 5, it will affect the hydrogen bonds. if I give this pepsin 3, now this is the optimum pH for pepsin. So pepsin works at its most activity. So if I erase this all, let's talk what happened when we decrease the or increase the pH from its optimum level. So let's talk. Or increasing the pH denatures or decrease the enzyme activity. So what do I mean by EA? EA is just a short form for enzyme activity. If it is written on a graph, you can easily remember EA represents enzyme activity. Enzyme activity. This is shown on the graphs. I just wrote this in my sentence. Decreasing or increasing. If I decrease from 2 or if I increase from 2, the pH denatures or decrease the enzyme activity. What do I mean by denature? I told you it affects, when we change the pH, it affects the hydrogen bonds. What are hydrogen bonds again? Hydrogen bonds keep the protein molecules which makes enzymes together. So when the bonds are weaker, the enzymes breaks, the enzymes walls denature. If an enzyme have this shape, the enzyme would become something like this. This means that the bonds between the protein molecules that's keeping the protein molecules together is now breaking. It's trembling apart, it's flickering apart, so it denatures. The shape is not like this. If suppose this is a pepsin enzyme, I increase the pH from 3 to 4. This denatures the enzyme and change the shape of the enzyme. So what is the purpose of pepsin? Protein. It breaks down protein into smaller molecules. So this is the protein molecules. It has to fix over there. Can it fix over here? No. Why? Because this shape has changed. This shape was the shape that this protein molecule can go and settle down. This substrate, just suppose this is a substrate and it wants to break down in its product because the body needs it. So this enzyme cannot break this substrate. Why? Because the shape of its active site that breaks the substrate is changed. It cannot take its purpose. It can break, but how? Why can it don't break? Because how this substrate, a square substrate, for summarize or gets into this irregular shape. Do you have any sense? Alright. If I write this, change in active site greatly impacts enzyme activity as no substrate can settle in the enzyme active site. I hope you all understand what I meant to say in this sentence. Change in active site. This active site has changed greatly, can affect the enzyme activity. Now this substrate has to wait or depend on another pepsin enzyme. This substrate has to depend on another enzyme which is already busy. So this greatly affects the enzyme activity as no substrate can settle in the enzyme active site. This enzyme is permanently denatured. I hope you all understand this whole topic. So thank you for watching my video, guys.